I hope you're ready for this one because we are now entering into probably what I would deem the two most difficult lectures of the entire semester. Now, I don't want to do that to scare you too much, but at the same time, statistically, these are the lowest scoring quizzes in any biology class, not mine, just any biology class, because now we're getting into the processes of how everything we've learned up to this point start applying, especially when it comes to the two major processes of how energy is first captured and then undergoes metabolism. So remember that these two lectures, this one and the following one, are all about how living things use energy. And we talked in the last lecture about how the cell maintains homeostasis, sometimes with entropy, sometimes against entropy. But we also talked about the laws of energy and how when energy is transferred, you never always fully transfer it, which is why life on this planet is completely dependent upon one process alone, which is photosynthesis. Without this process, you and I would not be here because we don't have the capability of renewing any energy. We only can use up the energy that is being captured by these photosynthetic organisms. So this really gets also into ecosystems and why it's important that we maintain stable ecosystems because of the chain of food that goes from these photosynthesizers up through the heterotrophs, which are you and I and all the other organisms that depend upon them. So this lecture is primarily going to look at how any photosynthesizer, but we're going to focus primarily on plants, how any photosynthesizer is able to capture sunlight, which is kinetic energy, and turn it into covalent bond energy, which biologically is potential energy. So the word photosynthesis, photo means light, synthesis, like we think of dehydration synthesis, is the storage of that energy by covalently bonding atoms together and forming larger structures like carbohydrates and fats and proteins and the like. So, plants aren't the majority of the photosynthesizers on our planet. In fact, they're the minority. The majority are aquatic photosynthesizers like algae and other such. There are even some microorganisms like bacteria that, though they don't have chloroplast, still can harness solar energy via other proteins and mechanisms. So we're primarily going to look at chloroplasts in this lecture, but just be aware that there are many, many, many different organisms on this planet that can undergo photosynthesis. Plants, protists, bacteria, these are kind of the kingdoms or domains that have organisms that can undergo photosynthesis. Animals, on the other hand, and fungi, we can't. We don't undergo any type of photosynthesis. We're the consumers and the decomposers. So the only way in which we can get our energy is by first allowing the autotrophs, which are the organisms that capture sunlight, harness that energy, and then we can get it biologically. Now, there are some organisms down in the deep recesses of the ocean that can live off the energy that comes through these thermoaquatic vents, but those are very few and far between. So we're primarily going to focus on the fact that all life is dependent upon photosynthesis to survive. So, little chemistry for you. The overall process of photosynthesis is as follows. And even this isn't the full reaction, but it's a simplified version of this reaction. Basically, any photosynthetic organism uses carbon dioxide, water, and then light energy as its source of kinetic energy. Remember, this is an exergonic reaction that comes from the sun. So this is the part of the reaction that actually supplies the kinetic energy. And then it uses that energy to covalently bond atoms together to form larger structures. This one right here being glucose. And then oxygen is also produced. So plants and other photosynthetic organisms generate oxygen in the process of consuming carbon dioxide and water. In fact, we'll show today that water and oxygen have a direct relationship with one another. The oxygen which you and I breathe and is in the atmosphere actually comes by the plant's ability to split water into hydrogen ions and oxygen gas. The oxygen from the carbon dioxide and then the carbons themselves that goes into the actual food making process. So we're going to dissect this overall process down. But there is going to be a question, very simply, that will test you on your overall understanding of this process, meaning what goes in, what are the substrates, which is carbon dioxide, water, and light. We won't worry about the numbers here, okay? So don't worry about total amounts. This is not even fully chemically balanced. The products are glucose and oxygen. Those are the two products of this overall process. Like any metabolism, there's always enzymes at every step of the way. But most of those enzymes we're just going to ignore. There are a few, however, that I'm going to make mention of because they play a role in understanding certain evolutionary mechanisms as well. Now, before we can talk about the actual structure of the organelle, the chloroplast, which is responsible for the majority of photosynthetic activity on our planet, we first have to look at sunlight as an energy source, because energy, as we've talked about, remember, comes in a wide variety of manifestations. Well, pure energy, like photons of light, can actually have higher or lower strengths, meaning they're more energetic or less energetic. So let's look at what we call the electromagnetic spectrum. You and I, the biology of our eyes, can only detect or observe a very small fraction of the amount of energy that actually exists in our universe. Notice that all of these are all different wavelengths of energy. Now, when we say wavelengths, when we measure them, the distance between the crest of these waves of energy ultimately tells you how strong they are. And we measure that in actually nanometers, which is pretty small. The visible light tends, we can see, our eyes and our proteins are specifically designed to absorb wavelengths of light between 400 and 750 nanometers. That's what we call the visible light spectrum. Now, there are other organisms that can actually see other things outside of this, maybe ultraviolet or near infrared. In fact, this is where we look at heat signatures for near infrared and whatnot. And we use, like for your Xbox, it uses infrared and other things to ultimately distinguish who's where, in that. Let's look at the highest level energy wavelengths, gamma rays. This will not turn you into the Incredible Hulk. It will give you cancer and kill you. So gamma rays, which have the shortest wavelengths, are the most energetic types of waves that are generated. Well, the sun's always pumping these out, but thankfully, due to the Earth's magnetic field, as well as our ozone layer, all of the gamma rays are pretty much redistributed and don't reach us. And that's a good thing. X-rays too. Most of the X-rays that come towards our planet are scattered away and don't reach us. X-rays, as we know, are used a lot of times when we're looking at bone density, but too much exposure to X-rays will mutate your DNA and ultimately cause you to have cancer. So if the gamma rays are strong enough, I've heard stories of people working in nuclear reactor facilities where they were exposed to just a flash of gamma radiation and pretty much they were dead the next day. So you get a high enough dose of that and your cells break down because of the energy that's absorbed by them and you're dead. Ultraviolet. This is what our body tries to protect us against, UV radiation. Now this does reach us. This actually gets through our ozone layer and will reach our skin. And this also can mutate your DNA in your skin, which is why we usually want to wear sunblock, is because though our body will react and create proteins like melanin to absorb UV radiation, in the process of producing that melanin, we are being exposed to UV radiation and therefore have a higher risk of getting mutated DNA and therefore cancer, which is why too much UV radiation will cause skin cancer over time. All right. And then when we go to the upper end, microwaves. Why do microwaves heat up your food? Do you know why? Well, the microwaves are set at a very specific wavelength that water absorbs. So when you put your food into the microwave, all you're doing is causing the water to absorb that energy and become excited, thus heating it up, thus heating your food up. So that's really how microwaves work. is we can use microwaves for communication, as in some scenarios we do that, but in your microwave oven, so to speak, in your house, that's all it's doing is creating a wavelength that is specific for water. Water absorbs that, and that's what heats up your food. And then, of course, radio waves. These are the weakest of all of the energy waves. They're passing by you all the time. In fact, the longer they are, the more you'll reach them out, when you're in the mountains. For example, FM and AM, which one do you reach? Which one reaches you more when you're out away from the cities? AM. AM has longer wavelengths and therefore is not absorbed as much by the mountains and other areas as the FM radio waves are, which usually get deflected by most of the environment. So that's why AM typically reaches you. You're able to hear that for longer periods of time. Okay, so now that we've kind of gone over some of the basics, let's look at light. When you see something, you're actually seeing the reflection of that wavelength of light. So if you see my shirt is red, which I'm wearing today, this is the wavelength of light that you're seeing. Guess what? The pigments in my shirt are absorbing these other wavelengths of light. Same thing for green, same thing for blue. But let's look at the extremes. If you see white, it's actually the reflection of all visible light. So here, the whiteboard, the pigments or the material in which this is made of actually reflects all visible wavelengths of light, and therefore your eyes pick it up and interpret it as white. Guess what black is? The absorption of all visible light. That's why when you go out on a hot day, you'd rather wear a white shirt than a black shirt. Why? Because the black pigments, the pigments in your shirt, are absorbing this and causing you to heat up more than a white shirt would, because the white shirt is actually reflecting all of those wavelengths of light. All right, so why do I bring that up? Well, what color are plants that undergo photosynthesis usually? Green. So guess which color is being reflected by those photosynthetic pigments? It's green. So the blue and the red wavelengths of light are actually what are being absorbed as a form of kinetic energy by the plant pigments. Now, there are some pigments that are in the leaves that do absorb green, but they're minimal compared to the major pigment which absorbs blue and red blue and red wavelengths of light, which is why pretty much most photosynthetic organisms are green. Now, we asked the question, just to help you reiterate on this. Why is the sky blue? Most people think they know why the sky is blue, but most people really don't. Well, the sky is blue because as the light hits our ozone, the major wavelength of light that gets reflected in every direction is the blue wavelength of light. So this is why no matter where you look, wherever you look in the sky, it's always going to be blue. But then, why does, when the sun starts setting, why do we get orange and red and yellow colors? Well, as the sunlight is traveling through the atmosphere, and as our Earth turns and we see it across a larger amount of ozone layer, most of the blue light is being reflected away, and the only light that actually gets through the ozone are the red and the yellow and the orange. Okay, so that's why when the sun starts setting, we're seeing the light through more atmosphere, the blue light's being reflected away, and we're seeing the blue and the orange and the yellow. All right. So now that you have that little lesson on, now I will test you on certain dynamics of that of what we just went over as far as the reflection of light, and you'll see how I incorporate it in here as we go through the physiology of the plant leaves and the chloroplast. All right. So chloroplasts, as we discussed back in lecture 6 on the organelles of the cell, is the organelle that actually undergoes photosynthesis. It's the part of the plant leaves that are able to capture sunlight and turn them into sugars. Now there's two main areas in the chloroplast that are of importance that you should know. Now the chloroplast itself looks like this. It has a double membrane around it. Now this is a membrane-bound organelle, just like the other ones we've talked about. this actually has two membranes around it. Now inside, the fluid that is inside of the chloroplast, because we're dealing with an organelle and not a cell, we don't call it the cytoplasm. We call it the stroma. So the stroma is the fluid that is within this double membrane of the chloroplast. This is where sugars are made. So when we talk about when glucose and whatnot is made, that's in the fluid area. Well, in addition to the fluid, we have stacks and stacks of membrane, which we call thylakoids. So thylakoids, notice these are the green parts of the chloroplast. This is really what makes the chloroplast green. It's not the membrane out here, it's actually all of these stacks. Also notice that they're hollow. These discs have a fluid inside of them, and so they're not completely solid. They actually have some space inside, and you'll see how that works. Well, having lots and lots of stacks of these thylakoids increases the surface area. The more stacks you have, the more space and surface area you have to undergo photosynthesis. And that's why they're stacked on top of one another, is because if a light particle goes through here and just happens to not get absorbed by this one, it'll get absorbed by the next one, or by the next one, and so on and so forth. So it really increases the amount of light energy that these little organelles can actually pick up and absorb. Better than any solar panels we've created so far. Now within the membrane of the thylakoid discs are a pigment. And this pigment, the most abundant pigment that is in the membrane of the thylakoid discs, is called chlorophyll. Now there are actually, if I think off the top of my head, at least 6 pigments that can be found in the thylakoid discs. We're only going to talk about 2 of them. And in fact, we're not even going to talk too much about those. We're just going to mention them briefly. Chlorophyll is the most abundant pigment in any photosynthetic organism, which is why most photosynthetic organisms are green. Because if we look at the spectrum from which this molecule absorbs light, we find that it absorbs primarily blue and red wavelengths of light. And it reflects away green. So that's why most photosynthetic organisms are green. Now I say most because there are certain plants that have purple leaves and some almost black. I mean, they're really dark, where they're absorbing a lot more light energy. But there's a trade-off usually that comes from that. So green is reflected away by this chlorophyll pigment, but blue and red is absorbed. These are the 2 major wavelengths of energy that are absorbed. Now I'm not going to distinguish between A and B. We're just going to call it chlorophyll. So don't worry about different slight, because if you look at it, really each version takes on a different wavelength of light within that area. I'm just going to say blue and green. We're just going to, blue and red. We're just going to keep it nice and simple. But I do want to mention one other pigment. I never know what to call this, whether it's a carotenoid. I think the carotenoid is the proper way of saying it. Well, if you look at the first 5 letters, it looks like carrot. What color are carrots? Orange. So guess what? Carotenoids absorb blue and green wavelengths of light, but reflect away orange. Now, this brings us to a concept of why do leaves turn different colors in the fall? We'll see that later on this year. Well, it ultimately comes down to what pigments are still surviving in the leaf as the tree goes into hibernation. So as the days get shorter and as there's less and less light, some trees, not all, you know that there are evergreen trees and whatnot, but some trees will go into a state of hibernation where they'll essentially shed their leaves because the leaves aren't producing as much as they should be, and so it cuts the leaves off. Now as the leaves start dying, the first pigment to break down is the chlorophyll. But that leaves the carotenoids and other pigments like xanthophyll and other types of things behind. And when those get left behind, now the chlorophyll is broken down. It defaults to whatever the next most abundant protein or pigment is. And if it's a carotenoid mixed with some other pigments that usually last a little bit longer, then you're not going to be reflecting away green anymore. You're going to be absorbing blue and green and reflecting away yellow and orange and red. And each tree has its own composition of pigments, which is why every tree is going to have a different assortment or different color for its leaves, depending upon what pigments are in the leaves themselves. Now, eventually they'll turn completely brown as all of those pigments have fully broken down and whatnot. Oh, sorry. All right. So I will test you on the absorption of light by the pigments, because this is the key aspect of photosynthesis, in that it's the absorption of energy, not the reflection, but the absorption of energy that drives this endergonic reaction. So this reaction is all about absorbing energy and covalently bonding atoms together to form the organic molecules like glucose and, sugars and fats and proteins and such. So this first part's probably the most difficult of it all. So I'm going to go through this probably 5 times or so, different ways. So don't worry if you don't get it the first or the second or the third. If you don't get it by the 5th, then worry. But don't worry if you don't get it by the first or second time around that I go through here. All right? So the light reactions are these membrane-bound portions of the chloroplast, which we call thylakoids, that have the chlorophyll and carotenoid pigments embedded in the membrane. Now, as part of this process of harnessing the light energy, the chlorophyll and carotenoid pigments are arranged with other proteins in what we call a photosystem. Now, when scientists first discovered the photosystems, they discovered one and they called it photosystem 1. And they discovered that there was a second one that was distinct from the first one, so they called it photosystem 2. Well, then they discovered that photosystem 2 comes before photosystem 1, but they never changed their names, morons. So, when you look at because they do work in tandem with one another, photosystem 2 comes first, and then photosystem 1. Stupid. Shut a time machine, wring their necks. Okay, so it just goes to show that, they're like, well, no, we're not going to rename this one or rename this one anyway. So there are 2 types of photosystems, but no matter which one they are, they have 3 basic elements to it. The first is what we call the antenna pigments. What are the antenna pigments? This right here. Chlorophyll, carotenoids. Why do we call them antenna pigments? Because just like the antenna on your car picks up radio waves and through various processes translates that information, same thing with these. These absorb the energy. These have electrons. Well, all atoms have electrons, but these are specially designed to absorb the photons of light and energize electrons. So that's why we call them the antenna pigments. Okay? So both photosystems have what we call antenna pigments. Well, like any process of electrical current, like you would imagine what's going through the wall right here, we can't just tap into any part of that. So the way that the photosystem is arranged is all of the electrical energy, because that's really what it becomes at this point, is energized electrons. So when the photons of light hit the antenna pigments, they essentially energize the electrons, and the electrons take on more potential energy. As the electrons travel from protein to protein, they travel to a central core, which we call the reaction center. You can think of it as a plug. That's where we can safely harness the energy that's running through the walls here. There are special outlets where we can use that energy. Well, the same thing with the reaction center. The antenna pigments is this huge array that funnels all of the energy down to this core set of proteins called the reaction center. The last part of the photosystem is called the electron transport chain, or ETC for short. No, not that Heroes of the Storm character. So, ETC, electron transport chain. Well, guess what these series of proteins do? They transport electrons. That's what their name says they do. But this is where the two photosystems become different. Because in each of their electron transport chains, each one makes a different potential energy molecule. One photosystem makes ATP, the other photosystem makes NADPH. All right? So let me give you the gist of how this works. Here's the membrane of the thylakoid disc. Remember the phospholipids that make up all membrane-bound organelles and whatnot. Embedded in this membrane are the proteins and the pigments that are necessary for each photosystem. Well, here we have photosystem 2. So here's the order of operations, meaning step-by-step process. Sunlight is constantly bombarding our planet. Well, when the plant it gets hit by a photon of energy, which is blue or red or green or whatnot, the chlorophyll, which is specifically designed for different wavelengths of light, absorbs that light. That absorption causes the electrons to become energized. So you literally ramp up their energy and the electrons, have a higher potential energy. The electrons then get funneled down to what we call the reaction center, where they then get released by the reaction center into what we call the electron transport chain. As the electrons travel down the electron transport chain, they use up that energy. And I'll explain how they do it in both of these, but they use up that energy. Well, here's where photosystem 2 and photosystem 1 become connected, because the electrons that channel from photosystem 2 then go into photosystem 1. It's used up its energy, so it just gets re-energized by a new photon of light. And then it does the same thing. It goes down its own electron transport chain. So that's why these work in tandem with one another. Photosystem 2 funnels electrons through its electron transport chain to photosystem 1. Photosystem 1 funnels its electrons through its electron transport chain, which then get picked up by this energy molecule. So let's talk a little bit about the different energy molecules that they make. Here's where it hits the fan. So, okay. Photosystem 2 makes ATP. Just, if you can't remember anything else, this is the main point. Photosystem 2 makes ATP. Photosystem 1 makes NADPH. Okay, just hammer that into your head. And if you don't get this next part, you're not screwed. But okay, so let's talk about photosystem 2. How does it make ATP? You'll begin to see why we spent so much time on the modes of membrane transport in the previous lecture, such as osmosis and facilitated diffusion and active transport and so on and so forth, and simple diffusion. They all come into play right here. All right, so the electron, as it travels down the electron transport chain, gives its energy and allows these proteins to pump hydrogen ions from the stroma into the thylakoid disc space. What do we call that? When you pump a molecule from a low concentration to a high concentration using energy, what do we call that? Remember, there's simple diffusion, there's facilitated diffusion, there's osmosis, there's active transport. Which one of those is it? Low to high, against entropy, using energy. You better know these for this week's quiz, for quiz 7. Active transport. Okay, so active transport requires energy. Why? Because you're doing what doesn't naturally occur. You're taking molecules, in this case hydrogen ions, and pushing them against their concentration gradient. You're blowing up the inside of this thylakoid space like a balloon, but you're just filling up with high concentration of hydrogen ions. So that's what this does. This is essentially a proton pump. This pumps protons, because that's all hydrogen ions are, pumps protons into the thylakoid space. Okay, well they're trapped in there now. And now you have this high concentration of hydrogen ions. Well, remember we said that high concentration is actually potential energy. So guess what? On the flip side of this thylakoid membrane, we have a special enzyme called ATP synthase. This enzyme opens up a channel in the membrane and allows the hydrogen ions to flow from a high concentration to a low. Remember, that occurs naturally. This works with entropy. What do we call that when molecules go from a high to a low through a membrane protein? Facilitated diffusion. So here we have active transport that requires energy. Here we have facilitated diffusion that occurs naturally. That occurs just from a high to a low. Well, remember we said that movement is kinetic energy. So when these hydrogen ions are moving through this protein, that kinetic energy is used to covalently bond this phosphate to the used up version of it, which is ADP, and that's how ATP is made. Guess what? This enzyme is also found in mitochondria, the same enzyme that cyanide blocks. The same enzyme. That's why cyanide kills you, remember? So this enzyme right here that allows hydrogen ions out and uses that energy to covalently bond the phosphate with ADP to make ATP. That's ATP synthase. That's how ATP is made. Okay? So basically it blows it up like a balloon by pumping hydrogen ions in here, and then as they go out through facilitated diffusion, that kinetic energy is turned back into potential energy in the form of ATP. We have a name for this. It's a very fancy name that tells you what I just explained. It's called chemiosmotic phosphorylation. Okay? Didn't make that up. So, what's chemiosmosis? This right here. Just pumping hydrogen ions into here against their concentration gradient. What's phosphorylation? It's when you attach the phosphate here to make ATP. That's all it is. So, you won't have to explain that back to me. But you do need to know this. Chemiosmotic phosphorylation makes ATP. Okay? Chemiosmotic phosphorylation is this. What I just explained to you, that's how ATP is made. This is exactly how it's made in our mitochondria as well. The only difference is it's not made with chloroplast, it's made with mitochondria. Okay? This process, chemiosmotic phosphorylation. All right, so let's move on to the next step. This one's so easy. As the electrons go into photosystem 1, they get re-energized because they used up that energy when they pumped hydrogen ions here into this membrane space. So they get re-energized by a new photon of light. Now when these electrons travel down the photosystem 1's electron transport chain, they don't do anything special. They just get picked up by what we call an electron carrier and get turned into what we call NADPH. That's it. See how simple that is? So they don't lose their energy. In fact, the electrons keep that energy. And when these NADPs pick up the electrons and some hydrogen, then they form what we call an electron carrier. Notice ADP, these are almost identical to one another. Okay, so they're both energy molecules. You won't even have to worry about what the shape of NADP is. Okay, recap. Photosystem 2 makes what? ATP. Photosystem 1 makes? Good. What's the name of the process that photosystem 2 uses to make ATP? All right, see how easy this is? All right, that's the hardest part of it all. Okay, one more thing to go over. You're like, oh, hell. Okay. The electrons here are actual matter. Remember, atoms are made of protons, neutrons, and electrons, right? As electrons go through this process, they're being consumed here at the end. The NADPH is picking them up and pulling them away. So then you have to ask the question, well, where does this get more? Because they don't just pop out into existence. The electrons have to come from somewhere. Here enters water. And this is why your plants need a substantial amount of water to occur. Guess what? As you water your plants, what is water for in this process? What is the whole purpose of water? The plant wants the electrons that hold the hydrogen and the oxygen together to form water. It doesn't really care about much else. It just wants the electrons. So guess what? Some of the energy in photosystem 2 splits water, breaks the covalent bond, and steals the electrons that the hydrogen and the oxygen were sharing with one another. So as it steals those electrons, those then refuel this process. That's why if you don't water your plants, this doesn't occur. Because without the electrons to go through this process and make ATP and then get over here to make NADPH, you don't make sugar. You don't make anything. Here's also where the oxygen that you and I breathe comes from. When plants give off oxygen, that comes from the water we give them. They're literally from the water we give them. Now it becomes oxygen. Well, let's talk about the two other processes we discussed. As oxygen builds up inside the thylakoid space, how does it escape? You remember? What's the name of the process where molecules go from a high to a low concentration through a membrane?
Active transport.
Yeah. High to low works with entropy. Simple diffusion. So all that happens is as oxygen builds up, it just leaks out and then leaks out into the air. So as it builds up inside, then it diffuses, simple diffusion, out of the thylakoid disc, out of the chloroplast, out of the cell membrane, and it goes into the air. That's it. Water, on the other hand, as it's getting used up and you start having less and less water in the thylakoid space, how does water get in? Through? No? Osmosis. There you go. Osmosis just draws more water in. As there's more water on the outside than on the inside, as this is going down, then water just comes in and keeps this process going. We've talked about each one of these now. Osmosis, simple diffusion, active transport, facilitated diffusion. See? It's all there, all relevant. All right, so. Now, up to this point, the plants just made some ATP and NADPH. But remember, those are short-lived energy molecules. They don't last very long. They need to be used up right away. Well, thankfully, there's a reaction in the stroma that surrounds the thylakoid disc that is more than happy to use that energy up. We call it the Calvin cycle, named after the scientist who discovered it. That's why it's capitalized. So the Calvin cycle is essentially glucose synthesis. However, I want to point out that the plant doesn't just make sugar. As we've learned that simple organic molecules can be assembled in a variety of different ways. Well, some of the products of the Calvin cycle make sugars, absolutely. And from there, remember that these are the monosaccharides that build up the polysaccharides, like cellulose, which form the cell wall. Or starch, which is the energy storage polysaccharide for plants. Or even some disaccharides, like sucrose, such as the fruit sugars that a plant would make. So, again, this is about not just making sugar, it's about making everything. Not just carbohydrates, but also amino acids, and fatty acids, and glycerol, triglycerides, and all those molecules, organic molecules that we use for food and energy. Okay? So, I just want to, though we're going to talk about glucose, make it clear that the Calvin cycle is really about making all organic molecules, not just carbohydrates. Okay, now this one can be difficult, but I'm going to simplify it down to three main steps. Okay, three fundamental steps. The first one is called carbon fixation. This is the process where the chloroplast will absorb carbon dioxide and take it from a gas state and chemically bond it to start forming the organic molecules. That's why we call it carbon fixation, is when you bind something and bring it from a gas state, say, to the solid state. Okay? But it can't just slap it together and form glucose right away. It actually has to go through a number of metabolic processes before it's ready to make a sugar. One of those processes that we're going to talk about is called PGAL, or phosphoglyceraldehyde, PGAL. is the shorthand for it. But phosphoglyceraldehyde, these are the building blocks for glucose, for amino acids, for triglycerides, for everything, really. You know, that's what I'm showing you right here. This G3P in this picture is the same thing as pGal. So this is phosphoglyceraldehyde. It just depends upon who's calling it what, but it's the same thing. So this right here is essentially pGal. Now, the third step takes the leftovers from this process and remakes an organic molecule that's necessary for this cycle. So let's look at this cycle and how it goes. Like I said, I'm going to skip almost all metabolic processes and just keep it simple, and we'll just look at the stages and what's going on. But there are a few key points that I do want you to know, because one in particular has to do with an evolutionary mechanism that plants have evolved to survive in hot and dry climates. We're going to bring this one up quite a bit, especially when we talk about evolution later on. So let's talk about carbon fixation. RUBP, which is called ribulose biphosphate. Remember that ribose sugar, that 5-carbon sugar we've talked about, the same one that makes up DNA and RNA, the deoxyribose and the ribose. Well, ribulose biphosphate is a 5-carbon sugar that's necessary for this process. Remember I told you can't just slap 6 carbon dioxides together with some oxygen and hydrogen and form glucose. It just doesn't work that way chemically. Well, this molecule is what we call the starter molecule. This is what gets the cycle rolling. So without this, you don't make glucose, all right? Well, here's how carbon fixation works. This 5-carbon gets picked up with the carbon dioxide by an enzyme and forms a 6-carbon sugar, not glucose, some other type of sugar that's not ready yet. And then it cuts it in half. Well, this is the only enzyme whose name you're going to have to know. You don't have to know any of the other enzymes. Now, the longhand name of it, don't bother writing this down, but the longhand name of it is ribulose biphosphate oxygenase carboxylase. Okay? The shorthand is Rubisco. Okay? So, Rubisco, nice of them to shorten that for us. Rubisco is the name of the enzyme. Okay? So Rubisco comes in, grabs the RUBP and carbon dioxide, covalently bonds them, and then cuts them in half. Okay? That's the first step. That's carbon fixation. The next step is what we call pGal synthesis. Well, these are unenergized carbons. So the ATP and the NADPH from the light reactions come in here and give their energy to these carbons. And that's what forms what we call pGal. Once those use up their energy, they go back to the light reactions and they pick up some more. They get re-energized. So that's what I showed you before, right here. The ATP and the NADPH that are made in the light reactions come here, give their energy, and go back. They come here, give their energy, go back and get re-energized. That's the connection between the two. Well, now that the pGals are made and are energized, now there's sufficient energy. Remember that energy of activation? Now there's sufficient energy for another enzyme to come in here and combine them and form glucose. Now, not all of the pGals are used to make glucose. Only like a sixth of them are. So what happens to the rest? What happens to 5/6 of these molecules? Well, they get turned back into RUBP. You may think, man, that's wasteful. Well, that's metabolism. So metabolism takes these remaining ones, turns them back into this 5-carbon sugar, and the cycle continues. Ultimately, 6 carbon dioxides come in, and a 6-carbon sugar comes out. That's the Calvin cycle. Don't worry about the rest of the metabolism going on here. It's metabolism, organic chemistry. It's beyond what you need to know for this class. The reason why I wanted you to know the enzyme that catalyzes the reaction in the Calvin cycle is because 5% of the plants on our planet have a big problem with undergoing the Calvin cycle when it's really hot and dry. But they have evolved a secondary additional pathway in addition to the Calvin cycle that allow them to survive in those very hot and dry climates. All other species, the 95% of other plant species, solely use the Calvin cycle to make sugars. However, there are a lot of areas on our planet that are very hot and very dry. Well, here's the problem. When the plant is undergoing photosynthesis, remember that right here, oxygen is produced in large quantities as the light reactions make ATP and NADPH. Well, normally, in normal environments, the oxygen is able to just easily diffuse out of the leaf cells through these little openings that open and close during the day. Well, one of the problems with desert plants is if they keep those openings in their leaves open for too long, not only do you get oxygen escaping, which is what it needs to do, but you also get water evaporating, which is a bad thing when you're in the desert. So they close those openings for long periods of time during the day so that you don't get a lot of water evaporation during this process. But that causes another issue because now the oxygen can't escape the cells. And as oxygen starts building up, there becomes a big issue with the Calvin cycle. Rubisco, that enzyme that uses carbon dioxide with ribulose biphosphate sugar, when oxygen starts becoming more abundant than carbon dioxide, this happens. We call it photorespiration. So instead of turning ribulose biphosphate into these PGAs and then converting them into PGALs and then into sugar, what ends up happening is the oxygen outcompetes the carbon dioxide because there's more of it. Well, the Rubisco enzyme can't combine a carbon there if there is no carbon, if there's just oxygen. So it splits it still, but in splitting it, this becomes unstable, this breaks apart, and you actually release carbon dioxide instead of capturing it. That's the complete opposite of what plants need to do to make their sugars. So this is what we call photorespiration, where this is a wasteful process when oxygen starts becoming too abundant in the cells. Now normally plants don't have to deal with this because they live in arid enough environments where there's enough moisture that they can release the carbon dioxide or release the oxygen readily enough that it doesn't build up. But like I said, in these hot, dry environments, this is an issue. So how have these desert plants overcome this problem? They use what is called the C4 pathway. Now here's a key point you have to understand. This doesn't replace the Calvin cycle. All plants use the Calvin cycle. Okay, let me reiterate that. All plants use the Calvin cycle. That is the only method that they can, that's the organic chemistry behind glucose, making glucose in organic molecules. Okay? But these desert plants have an additional pathway called the C4 pathway. Now the reason why they call it the C4 pathway is because the first step in this organic chemistry process creates a 4-carbon molecule, hence the C4 pathway. That's why the Calvin cycle is called the C3 pathway, is because it creates these 3-carbon PGAs during its metabolism. Okay? So what is so special about the C4 pathway? Well, there's many different strategies that different plants can use, but they all have the fundamental same organic chemistry behind the C4 pathway. And here is the trick. The enzyme that the C4 pathway uses to capture carbon dioxide doesn't care how much oxygen is present in those cells. So it doesn't make the mistake of trying to combine a sugar with oxygen like the Rubisco does. So in that scenario, what happens is these desert plants can close their leaf openings for long periods of time. Oxygen builds up, but the Calvin cycle just keeps going because instead of capturing carbon dioxide with Rubisco, they capture it with a different enzyme which doesn't have that problem with photorespiration. And then they just move it over to the Calvin cycle. They say, okay, here's the carbon that you need for the Calvin cycle. And they completely bypass the issue with the Rubisco enzyme.